Welcome to this latest video where I talk about the paper in the link below, which basically says the antibodies don't work. As always, this is a catchy title, so there's much more nuance to it. But what it talks about is the reproducibility crisis in the biological science, because a lot of the commercially available antibodies don't function as they should. So what happens, there's a lot of waste of time and resources due to these antibodies not working, which can be both, just a waste of money, but also in a lot of the cases you have to validate these antibodies for animal experiments. So there's also an animal welfare concern. So this is not a new problem. In general, in the 90s, people would produce their own antibodies from animals. So obviously this is a very time-consuming process. So what happened then, there was like a push towards commercial antibodies, which obviously, as everyone knows, they are expensive, but you save a lot of time in terms of like labor expenses. So nowadays there's over 7.7 million of those antibodies in the market from over 350 suppliers. So please note that a lot of antibodies, they might have different names, but they're actually the same. So there's probably less antibodies than that on the market. So in the next slide, I talk about how could you potentially solve that problem. You buy an antibody, it's like the same as buying something from the shelf, you expect it to work. So what are the authors of this paper propose? So they propose an initiative which is called Antibody Characterization for Open Science. So in short, I think the article mentioned it's pronounced as Icarus. And this is an initiative for testing of antibodies. And they test these antibodies in a very specific way and probably in a more detailed way than you would do it if you would get it from the lab. In essence, they test the antibodies by comparing a specificity in a cell line that expresses a target protein at normal biological levels against the performance in a knockout cell line. So in essence, what they're doing, because they're using this knockout cell line that doesn't express that particular target protein, you include a negative control. And this is far more effort, and it's definitely not something that you can do in any lab. Because of the inclusion of this negative control, it's a better measure of it, but it's a far more time-consuming process. So this is not something that you can routinely do this yourself. And this is what we'll talk about in the next slide, how you would normally validate these antibodies. But actually, through this initiative that a lot of companies are cooperating with this, to date, 73 antibodies have been removed from the market. So it's definitely making an impact, and both like the customers and suppliers are becoming more aware of this problem. So how would you normally test this in the lab? The first approach, the immunohistochemistry approach, is probably what you would do yourself. You would have like certain cells, and you have specific cells that express a specific target protein that you're interested in. The antibodies that you would have, they would label this target protein. You perform a Western blot, and you would look at the molecular weight to find the band that corresponds to the molecular weight that you're actually expecting. And also you would expect a single band. You wouldn't expect a mixture of them, because that would indicate some cross specificity. Another example or another method of how you can do this is using immunoprecipitation, where you have antibodies attached to beads and you have the protein of interest that's captured. Then you kind of take this out from the beads solution, and then you confirm using mass spectrometry, because you would also equally know what the molecular weight is supposed to be. However, in the previous slide, we talked about this different approach where we include the negative control, which is something that you don't particularly do here. And if you use this knockout cell line, so this is what this Icarus initiative talks about, they've tested in the paper, they tested 614 commercial antibodies, and well, maybe not surprising to me, but maybe to people who actually work with antibodies, is that 2/3 of them did not work as they were supposed to. And I have been expanding this initiative to test over 1,000 antibodies to date. So you can imagine that this is a very significant problem if you work with antibodies in your lab. However, as the title seems to suggest, the antibodies don't work. That's definitely not the case. There's many well-functioning antibodies, well-characterized antibodies on the market, and this is what I'll talk about in the next slide, where to go next. So where do we go from here? First of all, we need to take this as a bit of caution, because if you are working with a very specific cell line, a very different protocol, then these experiments might not map one-to-one. So even if they might find in this database that the antibody is working very well, then if you're working with a different protocol, that might not be the case. So reproducibility and checking things yourself is always key. Now, another thing that has been proposed, so there's more things besides this Icarus initiative, is to use resource identifiers in journal articles, because a lot of the time when you read papers, you can't exactly pinpoint which antibody was used. Another initiative which has been very popular is a Cite app. So like you can cite your papers, you can also find the most cited antibodies. So that seems to kind of indicate the ones that are popular, they seem to be working well. Now, in my lab, we don't tend to work with antibodies so much, and the reason for that is that there's still a lot of animal-derived antibodies. So we do need to consider the principle of 3Rs, which relates to the use of animals in research. So even though there is recombinant antibody technology, there's still like a huge market for animal-derived antibodies, and it's not very clear that if you're interested in this topic, that you can actually find out how this is done. And another problem is that a lot of the same antibodies are sold under different names, so that leads to a bit of confusion. So in the future, it's definitely not the case that antibodies don't work. Even though I'm a polymer scientist, so I do have like a preference of working with biomimetic materials rather than antibodies. But it is like a call for if you're working in a lab, if you encounter problems with reproducibility, which is inherently a problem in the biological science, let's be honest, have a look at the antibodies that you're working with. So these are all really great initiative mentions in the paper, and hopefully we'll see more of this, but also we'll be interested to see in the next coming years to see more of, for instance, organ-on-chips or other biomimetic materials, like for instance aptamers and polymers that can start replacing these antibodies. Thanks for watching. If you're interested in other videos on this topic, do have a look at our playlist, Bio-Inspired Materials.